Thanks, Alex. Thanks, everybody. And thanks again, Alex, for the kind invitation. And thanks, everybody. I know it's New Year's Eve, so thanks, everybody, for joining today. So I'm Orlagh Neary. I'm originally from Ireland, but I've been living in Seattle for over 2 decades, and I spent most of that time at Microsoft. And I just want to add a big, massive disclaimer. I'm not a physics guru. I'm not a scientist or a healthcare practitioner. I'm simply just a curious fangirl of quantum. I've got a ton of curiosity of language, about meaning, and about how I've always been fascinated around how technology may be able to help us humans, understand the world around us a wee bit better and also each other. And so I think that curiosity eventually led me down many different career paths and more recently into quantum at Microsoft, but from a go-to-market perspective. I was super lucky to be surrounded with really inspiring scientists in Microsoft. And now, after leaving Microsoft in the summer, I took a wee bit of time off. I'm still taking some time off. And I'm through Quantum Links. I'm just helping scale awareness really around quantum computing, at both the opportunity it can create and also the risk it can create for us all. And I mostly target business leaders and the general public. So thanks again, Alex, for inviting me here today, different type of audience. And so I thought what I could do today, everybody, is just talk about what I think happens when curiosity meets compute, and how quantum, that convergence, I guess, of quantum and AI together are already starting to reshape, scientific discovery in healthcare and also life sciences. But first, I thought I'd share a bit of a personal story and jump into our time machine. Okay, bear with me, and bring us back to the 70s. And so back in the 70s, like medicine did not have the imaging tools that today we take for granted. Ultrasound in particular, it wasn't widely available. Data was really limited. Doctors often had to make decisions with really incomplete information. And it was around that time that my mom, who later actually became a diabetic research nurse, she actually experienced a miscarriage. And she was told then in the hospital when she had the miscarriage that the pregnancy was no longer viable. But my mom had this feeling, just a feeling, that there was still life inside of her. And she actually had no idea that she was carrying twins. There was no way, right, at the time for doctors to see what was happening inside of her body. There was no imaging. But because she trusted that feeling, she didn't move forward and have the DNC. And then 6 months later, my brother was born, Gavin, and who she said was a gift from the angels. And now I will say that he's the brother. I've got three brothers, and he's the one that I fight with the most. So I don't know how much of a gift he was, but no, he is. We're super close. But anyways, that's just a story of like her having to follow her gut instinct, right? Versus let's jump back in our wee time machine and look how far we've all come, right? In our lifetimes here, in my brother's lifetime, we have better imaging, we have better data. And as a result, we have better care, we have better healthcare. And so curiosity is what, asks so many questions. Humans took, time to ask questions, to ponder on what if, we did have different types of technology, and it was advances in compute and tech, I think, that expanded really what was possible for us. And it's then discovery that followed. And so, I think that healthcare and life sciences fundamentally, there are many things, they're very, very vast fields, but fundamentally they are discovery problems. Like in drug discovery, like for example, we're searching like vast chemical spaces, vaster than the universe itself. Biology itself is super probabilistic, right? Non-linear, so many different options, super complex. And, the cost of failure in all these worlds is, catastrophic, not just dollars, right, but time and lives. It can take, listen to my mom, a diabetic research nurse, it can take over decades and billions of dollars to bring a single drug to market. And the challenge isn't really, a lack of curiosity, a lack of data, a lack of intelligence, my goodness. The challenge really is how can we scale faster, and how we can navigate so many possibilities really at scale. And Alex, if you go to the next slide, that'll be awesome. And everybody, I've been playing around with Nano Banana on Google Gemini, so I thought I'd have a bit of fun with closing out the year. So here are some images courtesy of Nano Banana. So AI and has already helped, have significant impact, right, in helping us navigate so many possibility. AI today is already helping researchers predict protein structures, right, identify promising drug candidates, analyze massive, massive biomedical data sets faster than ever before. And I think or hope many of you on the call today already have heard of, what DeepMind AlphaFold was able to do. So they were able to predict the structure of, I think it was over 200 million, maybe be more, but I think it's 200 million proteins. And then what they did was they just didn't hold on to that data themselves. They made it freely available for scientists around the world to innovate on top of. And so I think that's an extraordinary acceleration of discovery. But the thing about AI, and I love AI, right? I use it every day in lots of different ways. I was involved in AI in the early days of Microsoft, so don't get me wrong, it's super powerful, and that's what's helping us advance today. But there are boundaries to what it can do. Like it learns from existing data that we have, right? And it really where I think it really excels is in predicting, right? In pattern recognition. But it does operate within the realm of what we already know, maybe what we already understand. Definitely helps us move faster, right? But it doesn't help us push further. And so I think, I'll throw a Star Trek reference in here that I love for the day that's in it. Like, I think just quantum really helps us go boldly where we haven't gone before, where we haven't been able to explore before. And so, Alex, if you move a few slides down, I think quantum on the next slide, Alex, thanks. Not that it matters, the visuals are just fun. But I think quantum really has changed the nature of how we explore. AI helps us search smarter for sure, but quantum help us explore differently because it's a new paradigm of compute. And I know, Hanah, you're probably going to talk about this way more from a scientific and technical perspective. But anytime I read stuff about quantum, I get triggered when it just talks about speed because it's not just about speed. It's thinking differently and different workflows and everything. And that's why like these different methodologies, different ways of working are really promising for chemistry, for material science, and for biology, where our classical way of compute and simulating things just hits a dead end. And so I'm really excited about the possibilities. Looking forward, Hanah, to you talking more. And so I think like up till now, like we've been able to ask from us curious humans, we've been asked like, can we do this? Can we do that? Can we speed up discovery? Can we find better molecules, right? Can we find better drugs? Can we reduce, if I think about my mom in diabetic research, can we reduce the number of failed, experiments, right? They're all super important questions in terms of can we, right? But quantum helps us go from can we to what if, right? I love it. And so like what if we could simulate more molecular interactions? Um, what if we could simulate how nature actually, works? I'm really fascinated by that, right? I was talking to somebody recently and I was asked, like a physicist, I was asking them, like, you've given your lifetime to exploration, like why? And, he was deeply religious and he said, quantum actually makes me feel closer to God. There's that paranormal, aspect to it. Now, I'm not here to debate religion or politics or anything, but quantum just opens up a new world of discovery for us. So I thought, what I could do in the next slide, Alex, is just share like 5 scenarios that I am excited about that I think this convergence of AI and quantum enables. So first off, healthcare ops. Okay, not super sexy, but if you think about it, it is actually it's the place where care starts, right? So, what if healthcare didn't feel like a constant bloody emergency? What if hospitals could actually see the bottlenecks before they actually happened, not after, patients are lying in hallways on trolleys waiting for beds. What if staff was less burned out, And so I think hospitals are some of the most complex systems that we run. Many of my cousins are nurses, actually. unfortunately. I didn't follow in my mom's footsteps, but they did. But there many of them are working shifts, this New Year's Eve that will last forever. But today the decisions that healthcare operations, it's super reactive, not necessarily as proactive. Sure, it has advanced, but still reactive. And I love some of the work that I saw that a quantum company called D-Wave is doing. They're working on optimization problems like schedule optimization, resource allocation optimization using quantum algorithms, right, to help hospitals figure out, the challenges to some of their optimization problems. And so quantum inspired compute is already helping us today make healthcare ops run more efficiently. The other one I love is disease detection, right? Like, who on the call today does not have a disease or know somebody who has a disease? But what if, right, we could detect the symptoms before, the disease even actually happens, like before anybody's in pain, before there's a massive crisis, and before there's like late stage, intervention. Like, think about cancer when, many unfortunately, family members of mine, detecting cancer in when it's stage 2, stage 3, and even stage 4. Like, what if we could detect it way earlier? And so I think, what I've got really interested in beyond quantum compute is also quantum sensing. And so there's researchers around the world that have published work. In particular, I saw one recently around the University of Nottingham, I think it was, where they've published work on quantum sensors that can detect like incredibly, faint, tiny little biological signals that are, completely lost, but they're able to detect them. So if you can detect these signals, right, the possibility is there for us to catch problems earlier and also less invasively. And so I think that's a fundamental shift from reaction to prevention. And there's a lot of great work happening today already in this space. So it's not like science fiction. It's not 5, 7. You can debate how far out quantum compute actually is. It's already tangible today. The other one, of course, I love is drug discovery. And so, think about it. Like, what if discovering new medications didn't take 10, 15 years and billions of dollars? And what if we could narrow down the promising drug treatments much, much faster? Drug discovery is incredibly slow. I think I talked about it a wee minute ago. It's expensive, full of trial and error. And the hardest part, right, about drug discovery isn't making the molecules. It's about understanding how they behave and which ones are worth pursuing. Unfortunately, this is where I had some deep exposure in Microsoft through a platform that Microsoft launched just before I left called Microsoft Discovery, where it's combining, right, AI, HPC, and quantum inspired chemistry models to really help researchers explore molecular behavior in a much faster and more efficient way. And so, again, quantum is not replacing labs, doctors, scientists at all. It's just helping us ask better questions sooner and sort of reducing the guesswork. The other scenario that I'd love to talk about is brain health, right? So, brain and mental health conditions are one of the most widespread and the least actually understood, I think. And they're massive challenges, right, that we face. But unfortunately, we still diagnose like many mental brain conditions based on observation versus, like behavioral observation versus actual biological signals. And again, this is where I think quantum sensing excites me a lot, folks. It's what if we could truly understand the brain, not just observe that behavior, but see the clear signals of what's happening underneath. And there's promising research work that's happening in this space as well. And that opens the door to earlier diagnosis, less invasive monitoring, and just a better understanding, right, of mental health. And if we get all of that right, there's a lot of work going on there. There's so much more opportunity out there, like education, productivity, aging, and just quality of life in general. And the last one, a bit like healthcare ops, not as sexy really as maybe the other three that I really love. It's also data privacy, right? Because at the end of the day, health and life sciences, they're deeply personal, and there's a ton of personal data there that is long lived. And data that is stolen today can be encrypted or could be encrypted, in a few years' time as quantum compute power advances. But fortunately, there's a lot of work. Again, love all the scientists that are I have the joy to have in my life that are working on post-quantum cryptography and really helping to protect the sensitive data against future quantum threats. And so that's work that's already happening today to help us protect that data. And it's where we can all take actions today. And so, Alex, if you go to the next slide, please. Like I was talking to Alex just before this call a wee bit, like about how, I certainly, get wrapped up in the research, the technology, about what quantum AI can do. But at the end of the day, what the convergence of quantum AI gives us is time, right, and hope. It's time to diagnosis, time to treatment, time given back to patients and their families. It's about trust as well, right? Like, trust is key in science, in medicine, and in the systems that we're all part of. And it's about the outcomes. It's not about innovation just for innovation, although I kind of like some of that too. But it really is about innovation that genuinely improves people's lives. And so my mom, like I referenced earlier, she was a diabetic research nurse. She spent years working with patients, running trials, collecting data, often known that the impact of her work wasn't going to be, realized for years to come. But what she carried with her, right, every day, and she talked to me about her patients as well, was hope that the research that they were doing was going to lead to a treatment in the future. The hope that science, right, like if done carefully, responsibly, and compassionately, could really change their lives. And, she herself actually, she's got Crohn's disease. So, and that the discovery of her Crohn's disease didn't happen until much, much later in life. And, whilst our family, we all call her jokingly the Duracell bunny because she's got so much energy, I think back to, what if some of the technology advances that we're going to see, like particularly in quantum sensing, could have helped detect her disease much, much earlier. Like she could have had a much better quality of life. Like she has a great life, but it could have been so much better. She could have been living with less pain. Um, and so then on the right are my giants. They're my children, 22 and 18. But you know what? Both of them, when they were 12, 13, they were both weirdly diagnosed with retinal dystrophy, right? Which is a genetic condition that can one day lead to blindness, like legal blindness. It's basically tunnel vision. And today there's really no cure for them. They can't detect the mutation of the gene that caused the dystrophy, but there's research and there's progress. And I wasn't working in AI when both boys were diagnosed, and already because of AI, there has been more detection of the mutations, but unfortunately not their mutation. But what the convergence of quantum AI gives many of us is hope, right? Hope that advances will be made that will one day, help slow down the dystrophy or potentially, be able to manipulate the genes to be able to cure it. So my hope, is that our future is brighter because of quantum and AI. I'm a pretty optimistic person. I don't tend to dwell on the dark side and all the risks, although we shouldn't have our head in the sands. We should be actively working to secure our data against quantum attacks in the future. But I really think, like, and for my wish for the new year, like it is Happy New Year to everybody, is that we continue, that everybody out there who is in the research world, who is a scientist, like you, it takes so much persistence, right? And you've so much curiosity, and there's a lot of folks that don't believe in you or don't believe in us, that say no or don't fund or whatever. So my wish, my hope for 2026 for all of us is that we stay persistent, we stay curious longer, that we give ourselves enough time to sit with the complexity, we leverage the tools that enable us to drive the discovery forward. Because curiosity, it isn't a luxury. It's how new treatments are going to be found. It's how new time is going to be found to give back to folks. And so I hope we can all imagine responsibly together. Maybe there's going to be new scenarios in the future that are going to excite me, but I'm hoping that we can discover those together. So I think that's it. It was just a little tee up of from my perspective, a non-physicist, a non-scientist, what is exciting me most about the possibilities ahead of us with quantum and AI. So thanks, Alex, for the invite and for letting me share my musings.
Thanks. So yeah, so what exactly? Okay, so quantum machine learning is a very broad topic. It's a lot of people like to hype up machine learning and quantum as being um, something that will hopefully improve things like medicine, healthcare, all of these data-centric things which have huge amounts of data. And so we're trying to figure out ways to um, keep that data intact without truncating it in some way or another. So why is this important for a rare condition like Perry disease? So what is Perry disease? Perry disease is very similar to Parkinson's. It is actually driven by mutations that disrupt the dynactin-dynein microtubule transport effect. And so what you typically see is a systematic decline in patients, and it's a very acute, it can be considered a very acute form of Parkinson's. Next slide, please.
Okay, just a moment.
Yeah. So as I mentioned, Perry disease is driven by DCTN1 mutations. And so realistically, DCTN1, there are no existing therapies for that gene, which again makes people's lives with the condition extremely hard to deal with, because the only medication that they can take is L-Dopa, which is normally for Parkinson's, but in this case, because it's a very severe form of Parkinson's, it is not it's not unfortunately able to be treated in the best way possible. Next slide, please. And so there are some challenges currently in discovering these genes and discovering new targets, new pathways, specifically in rare disorders where you don't have a lot of data. And so some of that is in part due to what's called guide variability. So guide variability is essentially an RNA which almost acts like a messenger between two CRISPR cells of CRISPR genes. And so what can happen is if there is some noise in the system, you can get a lot of false positive, false negative cell pluripotency of cells. We also have batch effect, donor effects, specifically from those stem cells, from those pluripotent cells, which I'd mentioned. So because of mismatching effects from donor to donor, we sometimes get compatibility issues. We get correlation, causation doesn't really reflect actual rescue mechanisms of the gene itself. And then finally, we have overfitting risk computationally. So classical methods have a very low n, so a number, dimensionality of a feature space, but a very high resolution of data, so very deep levels of very sort of the data becomes very compact and very hard to isolate. Next slide, please. So the way that we set up our CRISPR setup was pretty mundane. If you know CRISPR, if you know how it works, essentially what we're doing is we're doing target space decomposition. So the first thing that we do is say we have some prior. So we have a genome, let's call it T, right, or tau, and we build a union of maps of various molecules. So in this case, DCTN1, dynein microtubule, vesicle, dopaminergic, stress. So all of these target molecules are in this target space. And so when we unionize all of these, we end up with a cardinality constraint, which we ended up getting between 10 to the 2 and 10 to the 3 molecules that were in our target space. And once we had that amount, we then set out to map those targets using a library. So we set L to RNA1 all the way to RNA-N. And so L in this case is just mapping the target that that the perturbation that the CRISPR is perturbing. And so this actually is important because when we look at cell level data, perturbation identity G is also going to be discussed, which is where we conglomerate all of this library data and be able to more closely inspect L as a library. Next slide, please. So so I briefly described what's involved. So we're going to go a little bit further into this. So perturbation operator. So in order to perform perturbation within CRISPR, we have to have certain operators. So you're probably familiar in CRISPR, we use something called dCas9, which is a gene that can act with CRISPR to help isolate certain, mammalian cell lines that are important. So in this case, the operator choice that we use, there's two types that we use in our set. So we have CRISPR-A and CRISPR-I. And what S and the set S is essentially activating. It's selecting activation versus interference. So A stands for activation and I stands for interference. So computationally, it's just going to flip the expression of whatever is happening within that CRISPR expression. So yeah, so as you can see, all we're doing is flipping. Most of the equations are intact, except we're saying that J, in this case, is, let's call J our guide RNA. It's targeting gene GJ, and the perturbation is basically going in a positive direction or a negative direction, right? And so this PYDOG here is the causal object. So it means that we set perturbation, an identity to our downstream score. So whatever we have as a distribution, it means that we will essentially store that as a list of contrasts following that S set here. So we're downstreaming those distributions. Next slide. So what we're doing here is lab automation. So again, a lot of people may not be familiar with how lab automation works. Lab automation is majority of the time anything that can be done to a cell in the way that it can change various factors which could improve its growth or could improve its ability to thrive in a given condition. So lab, in this case, the OpenTron system uses, it is a lab automation robot. It is a robotic tool to perform liquid pipetting. So it will it will sort of pipette certain amounts of cell into different petri dish, into different wells inside petri dishes. And so what we're doing here is sort of going from plate to plate. So you can see we have in the picture I showed 3 plates. So what we're doing is essentially we're going, okay, we're pipetting once, we're pipetting twice, we're pipetting 3 times. That's sort of the normal standard way of using this device, which is you pipette multiple times. And so with lab automation, we're doing plate to condition mapping. So what we can do is say, okay, let's think of W between 1 and W as wells, which I mentioned are those plates. And so what the robot does is it implements PW, so it guides in that L, that library, with a certain condition. And so this condition is then reproducible in a certain function, right? So we have a function NW is equal to N0 for L times C. So the function is then for all L times C for all W, right? And so again, we see here that's also in the controlled cell initialization stage. And then we do per cell perturbation. And so what happens here is each cell I is carrying a perturbation labeled GI. So any perturbation between 1 and K will get labeled with an index I, with a perturbation identity. And so what K ends up equaling is the whole library of that G sub I indices. So that's what we get as labels. And then barcode coupling will be when we have B sub I is a tag associated with that with G sub I. So it's a label within a label. So now when we do sequencing of the DNA outside of the cell, so the perturbation cell tags, which were G sub I, are now getting data labels for their RNA sequences, which are called B sub I. So that gives us phenotype, that gives us transcriptome, all of that kind of stuff. Next slide. So within this machine, we have what's called temporal control. So temporal control is about how we can optimize the machine in such a way that temperature, things like temperature, things like media for growing these the cells, things like magnetic fields, for separating plasmids. All of these things, these variables are controlled in a certain way. So in this case, temporal batching just means through space and time, we're just looking at one control. In this case, is the set of how many times are we running this batch repeatedly. And so we want to make sure that that is identical to W, which, as we said, W is the number of number of wells. So we want to make sure that the number of wells is consistent with how many times we're running these sort of pipetting transferring the cells between one well to another. Next slide. Oh, sorry, I missed something. If you don't mind going back, yeah. So the final thing is readout. So X in this case is the final readout. It is a matrix, and then Y is the phenotype vector, and then G is the intervention labels that we previously assigned. So everything here, essentially, we're getting an RNA sequence at the end, and then we're getting a phenotype. And so that phenotype tells us, is this a functional phenotype or is it a non-functional phenotype? If it is a non-functional phenotype, that gets delisted from our final feature matrix. Next slide. So where does quantum come into the picture? So what exactly are we using quantum for in this case? Quantum computing has a lot of different modalities. The modality that we used in this specific example was a photonic quantum computer. And the reason we use that is because it contains what's called a fixed nonlinear feature map. So what this means is we don't actually generate new quantum circuits. We only train classical linear circuits. Next slide. So a lot of this is very mathematical, but essentially what we're showing is for each perturbation condition, so let's start from the top. Input representation is set by a condition G. We summarize this this perturbation sequence vector as X sub G. And so index G in this case is denoting some guide gene, some aggregated group, depending on that pre-processing that we did with the lab automation step that we had in the previous slide. The next step, which is called quantum encoding, is where we start from the M. So we have a vacuum state, and so that's represented by X sub G, where we encode it as U ENC. And then we encode this unitary, injecting certain features, classical features, because as I mentioned in the previous slide, we're not training quantum circuits into this quantum computer. We're training classical models to fit onto this quantum device. Okay, so phases, displacements, squeezing parameters, all of these key tuning features of the computer are given as classical features only. They are not given as they are given as discrete features. Okay. We then do what's called fixed quantum reservoir nonlinear mixing. So what happens now is we have a reservoir and we're having fixed composition. So we're doing a combination of beam splitters, phase shifters, squeezers, which are all part of the photonic chip. And so we're then taking the superposition, we're taking the superposition of that entire reservoir to make sure that we're essentially quantizing the classical features that we injected into this state. Okay. The measurement readout that we get is m hat, which can be in this case photon number. It can be quadrature, so moments of correlation between those splitters. So we get a collapse of superposition. And so this gives us our measurement state, our sort of output. And so again, this is some function. This is some nonlinear function. And then finally, we get some scoring. We use a scoring metric, which is just standard convex ridge regression to show that we're not overfitting any of our targets. Okay, next slide. Okay. The first case that we looked at was transport adapters. So transport adapters are very important, specifically in that dynein, because as I mentioned, what is involved in peridisease is that dynein transport methodology in microtubules. So what we end up getting is a target subset. So we look at these words and numbers here are kind of a little bit confusing, but essentially they're just genes that we isolated out of this when we passed it through this quantum system. And we ended up looking at how well the causal effect, how well we saw, how good of a target were these different genes that we isolated from CRISPR. And we did see that there were a few, so the ones in green are actually relatively consistent with research. Some of the blue ones here are actually newer genes. So we found that it actually correlated quite nicely with what current research is showing, which is that these targets are acceptable targets to go after. And then we looked at cross-assay consistency. So we made sure that how viable are these targets long-term. And so we found, a lot of them to be very high up on that consistency scale. Next slide. We then did cross-validation. So to doubly make sure that we had a lot of our genes were, some of them were new. We wanted to make sure that those genes that were newer that were included were still viable options. So this was the output that we got. And it did show that some of the new ones have a very good gene out expression. So it means that they could be used as targets, but they're not as highly ranked as perhaps some of the other more well-known genes in peridisease. But they could be used at a push, particularly if you're looking at a different drug modality that you're not, very accustomed, you're looking at a monoclonal antibody versus some other stem cell based approach. So yeah, that's pretty much all. Oh, and then development. So we're looking at some other targets that we might be interested in. At the moment, we we want to see if we can embed some error correcting codes into our architecture to make sure that any noise that we get can help improve our runtime, and then also acceleration. So looking at maybe some larger data sets other than just 10 to the 2, 10 to the 3. We have some other ideas for combining different patient data sets and running this test this algorithmic approach with those enmeshed data sets as well. So yeah, feel free to reach out if any questions.
You can see my screen, right? Okay. So I'm going to briefly talk about, because I'm going to share the detail so you can see the content, because I just see that the quantum computing and especially in integrated AI is very complex. Or the math that actually that I did my PhD in physics actually in Johns Hopkins, and I did a lot of the work in mathematically oriented because I utilizing Einstein's the general relativity theory to understand the universe. And especially I studied black holes. It's 30 years ago. And after that, actually, I was industry and mostly working in engineering in kind of like all high-tech, deep tech, and health tech area. And based on my 30 years experience, actually, currently my focus is to make very complex science and engineering, like a quantum computing AI, to make it easy. So that's motivate to start to talk about like what the state of quantum computing now. And because as I told you that I my major was the science, and especially I have a deep dive into very deep level, math and physics. And after that, I have 30 years in industry, initially in university and also like a research laboratory, and then I just do more involved in the industry as a businessman and kind of like the innovation like readers in all different layers of the emerging technology. So even with actually I hesitate to talk about quantum computing because of the after my PhD, actually I move away from the more like a science than practical thing, right? So I hesitate something is not actually the mature enough to be in the commercial level, right? Like your ChatGPT is kind of like the after 30 years of the more than 40 years of the AI research and then in the commercial level, everybody can use, right? And then I think of quantum computing is not there. So I hesitate to talk about quantum computing for 30 years. But now I found that the situation changes because I noticed that there are a lot of the initiative. And even I found that during my search for the kind of like expert to invite as a speaker to my webinar, I found that several like experts already in working in the commercial level product and also like integrated AI. And also I found that there was even a lot of the investment, and even I found that in the Wall Street have a kind of like a NASDAQ has some special section about the stocks related to quantum computing. So that signals that it's something that the turning point now. So that's why I initially I deep dive into what the current state of the innovation and what is going on. And then I found there's very interesting things happening is, for example, that in the next week and then after the next week, there was a two big event. It's a CES in the Consumer Electronics Show in Las Vegas, and then a JP Morgan kind of event in San Francisco. Actually, both of these events signals the coming of the quantum computing. So that's the catch me that it's time to start to talk about it, and then way to actually make this more kind of like a commercial level, kind of like everybody's interest. So I actually as I mentioned that I move away from academic position for a long time. So I recently published several book chapters, but I never actually applied for some kind of like a peer review research paper. But I deep dive into this area, and then I found that this is very interesting things, and then I try to find that what kind of thing that I can contribute this very kind of like the significant moment. And then I delve into some research area, and then I finally get some result, and then I will share with you today. So I'm not going to tell about the detail, but I'm going to show some proof of concept live demos, so you understand what is meaning by the, like, for example, what kind of like a drug discovery is a process going on, because you already see that there are a lot of the math involved, a lot of the quantum computing happen, but I'm going to make this easy to understand in my understanding. So I will show that. As I mentioned, this is over maybe, I don't remember what is my peer reviews, the research paper as a published maybe more than 20 years ago, and then I submit my finding to the peer review journal. So it's under review now. So the title is Agentic AI Orchestrated Quantum Computing for Biomedical Discovery. So architect and then proof of concept study with the causal reasoning extensions. So it is about the 30 page. So if you are interested, I can share, then maybe you can start your own with this important area. So I'm not going to talk about this one, because what I motivate is to then I tried to find out the way to make kind of like a quantum AI to understand that even because the quantum computing infrastructure is not yet to fully deploy, so it is very expensive. And also kind of like there are some like a cloud service available, like Google Cloud, or kind of like AWS provides some quantum computing infrastructure access through the cloud service. So what I tried to do was without actually to utilizing this infrastructure, just to simulate them, and even with that simulation, then whether you can do a lot of the like important research. And particularly, I focus on kind of like very hard to develop the drugs that tried many different type of the approach before. So then I found that over past 2 years, I focused on specialized AI, agentic AI, and then found that the agentic AI can be a powerful tool to do this. So I just don't want to go detail, but here is agentic AI is act as orchestration layer. So for example, that it interpret goals, constructs. So it kind of like a co-filer with expert, quantum AI expert, to like expedite their process, right? So they have them to like make easier, to make like efficient way to they doing their research or their discovery. That's the key point. And then quantum computing and the causal reasoning as a part of this process. So the quantum computing is invoked selectively when classical approximation breakdown, and then physics ground validation materially improve the decision. And also, like, I use the causal reasoning to what the cause of the something have an outcome. And so I developed proof of concept to whether this framework that I developed can working. So I will show the 3 live demo. So I just developed demo to simplify that concept. So I'm going to show the 3 cases in live. So one is drug development, is KRAS G12D is very, very kind of like a history difficult target in oncology, right? So this to develop the drug for the treatment of this cancer is very difficult up to now. So I'm trying to show that the potential of kind of agentic AI orchestrated quantum AI can be a potential solution to develop to attacking this hard to find out in cancer. And other things that I'm going to show is it's kind of DNA based. More is it kind of like a previous one is they call traditional small molecule type of drug discovery, but it's more than that DNA based therapeutics. So it tried to kind of like find out that whether they can be find out some like a DNA based treatment like a breast cancer. And then I found that there is very strong potential can be utilized. And then finally, that I'm going to show is a rare disease. So in the rare disease, the problem is the not much the information is available to do or utilizing AI. So in this case, actually, I also utilize they call special type of the AI called casual AI. So the casual AI is not just to explain the process, but actually that they looking for what the origin or the cause of the something that the result in the outcome. So it's a special type of casual AI that actually currently the state of art. And then a lot of big companies and then innovative startups start utilizing this one to developing something, particularly that they are promising in the new type of the drug discovery. Okay, so I will show directly that demo. So that'll be the good to give you some snapshot what is mean by this, right? So I already did. So this is the one actually that I implementing agentic AI in ChatGPT, utilizing all kind of like prompt engineering. So it's not actually the commercial level, but it's very intuitive. And then it can make you understand what the process is going on and what will be the outcome of that. So in actual deployment, actually, you need some special type of the platform and devices and other kind of like a software you need it. But this is very intuitive way to actually utilizing prompt engineering to develop all type of the agentic AI. So it's kind of like what the agentic AI is doing is to understand, right? Because it try to understand what you want to do, right? Because they call perception. So it try to understand what we want to do. So it want to like design a small molecule inhibitor for KRAS G12 with improved like kind of like what you propose, like solubility and reduced hepatotoxicity. I'm not a drug developer, like a discovery expert. I'm just an AI expert, so I tried to make AI to understand. So this is understood by AI, not me. So and then it tried to go to for example, that it want to solve this one, and then it understand AI. This is AI, and then it understand, right? Because okay, so this is a special type of agent or planning agent as a clinical and mechanistic interpretation. So it tried to recognize what you want to do. So the parsing therapeutic object and extracting the characteristic pharmacokinetic and then safety content, and then process and then interpret design of it. So based on your input, it try to understand what you want to do, the goal, right? Clinical target and mechanistic intent and other thing. And then it understand what you want, right? You want to go next step. And then it try to understand like for example hierarchical task graph. So what kind of task like that you wanna develop? Starting from the like a lot of the like a small molecule candidate for this drug for this one, and then through this process that it try to for example target and also like molecular design, like uncertain detection, hybrid simulation, and multi-objective optimization, and cross-learning and synthesis translation. And then it based on this one, actually, it is a multi-agent system. So it tries as a specialist for each task. So it kind of like a target agent and molecular generator agent and quantum simulation agent, optimizer agent, and compliance agent, something like that. And then it tried to understand, for example, that what the current state of the research done. So it tried to understand what the current state of, and there was it look at the data source, that what kind of data source that it can look at it, and it tried the chemical and then pharmacological and genomic kind of all different type of the multimodal data that needed for this research. And then it tried to the knowledge graph update. And then initial molecular generation. So it tried to molecular generation alpha is activating generative, and then it tried to find out the potential candidate. So through the research, that it find out the total molecular generate is 50,000. And then first CRISPR screening offline and screening. And then it found that kind of like some qualification of the molecular general agent, the initial candidate library prepared. And then it found the next step, the quantum simulation. So initially the CRISPR methods to the research to find out the all potential candidate for the drug. And then utilizing quantum simulation to filter out. So quantum simulation agent alpha that evaluating confidence of CRISPR predictions. And then it tried to find out that from 2000 CRISPR screened the molecule, only 220 molecule flagged due to kind of like the quantum simulation. And then quantum method, it utilizing different type of the quantum method, and then find out the quantum output. And then try to find out the object optimization. So quantum assist optimization apply to fine-tuning functional group and also like a scaffold substitution and something like that. So this is too much kind of like in expert level. So, but I wish you that what it's going to do is to refinement. So the Agentic function is to through all the process of the utilizing classical drug discovery and also like classical AI, machine learning to find out and then also like develop like quantum way and then define it. So it orchestrates all the process of drug discovery, utilizing all the resources, classical AI, quantum and quantum AI, and so on. And then find out the total kind of like completed is 42. And then it proposed that the lab execution. And then finally, finally provide 12 candidate. So it greatly reduce the potential that to be developed utilizing in your drug discovery for this hard to cure cancer. So it really have to like your drug discovery process utilizing quantum computing resources and AI, and orchestrated by this agentic AI. So the agentic AI's function is to orchestrate all the process, understand and reason, and find out the way to like optimize and then filter out the best candidate, something like that. And then demo complete. Okay, so I don't have time to show all things. So DNA thing. So I already executed before webinar. So this is the same thing, right? Because it gives you all the process. For example, that this is case actually the DNA is a not small molecule. So it tried to find out where is, for example, here that it's the same thing, but you see that so it kind of like the agentic AI, the quantum guided autonomous oncology R&D system, and then it produces like try to understand what you want to do. For example, that it understand like what you want to do, and then select over the smart DNA Aptamer for HER2. I don't understand its disease, but actually it relates to like breast cancer, and then this AI understand the goal, and then task, and they try to understand kind of like what kind of like agent specialized agentic AI require, and then sequencing, so and then find out the quantum invocation. And then 15 DNA Aptamer Therapeutics reach kind of like a best candidate kind of thing. So basically this is the one actually actually this motivate I discussed with someone developing this one and starting from MIT, that actually they developing the purely kind of like a lab development, that they try to find out the key that some specific type of breast cancer, that this DNA actually the like intercept some like cancer cell, and then penetrate the cancer cell, and then integrate the RNA with this DNA and then try to kill. So I try to understand the mechanism, and then whether I can do not in the wet lab condition, but purely kind of like quantum computing and AI can develop this one, and I found that this is working very well. Okay, and then for the rare disease, I will show the just snapshot what I did. So in the rare disease, as I mentioned that this is case actually that require a lot of the reasoning needed. So for example, that here is one thing, the casual disease mechanism modeling. So in this case, actually, the system now construct express directed the causal graph. So it try to understand what cause of this one, right? So this is some specific, and then it in addition to the quantum computing, actually, they try find out what the cause of the reason. So and then I'll give you the final result. So final of rare disease therapeutic proof of concept. So quantum validate casual disease model, and then it tried to understand what cause, and then the proposing rational safety levels and kind of other thing, right? Okay, so I'll stop here. So